Hello everyone, I'm Nusra El-Maradi. I'm delighted to be speaking with you about COVID-19 coronavirus pathogenesis, clinical features, diagnostics, epidemiology, prevention and control in the first international online conference of pharmaceutical and medical sciences. This review was published in the Microbial Biosystems Journal, and it's our pleasure if you visit, download, and cite our paper. Throughout human history, there have been a number of pandemics of diseases, such as smallpox, shown here, and influenza. Even in this modern era, outbreaks are nearly constant. In our review, we illustrate some of history's most deadly viral pandemics, starting from smallpox to COVID-19. In this presentation, we will provide a brief introduction to coronaviruses, demonstrate and describe clinical features, epidemiological transmission mode, treatment and vaccination trials of COVID-19 globally and in Egypt. In December 2019, from Wuhan in China, COVID-19 was emerged as a new viral disease caused by a novel beta coronavirus, SARS coronavirus 2, which spread in more than 215 countries and 3 continents. At the time of this presentation, more than 23 million confirmed cases and 809,000 deaths and 15 million recovery cases of COVID-19 were reported globally. The highest numbers of cases were found in United States of America, Brazil, and India. And this graph showing the global total confirmed cases, as shown here, and total confirmed deaths. In Egypt, the first case of COVID-19 was discovered in 14th of February and reached to 79,000 total confirmed cases and about 5,000 total deaths and 65,000 recovered cases. COVID-19 spreads through direct contact routes, droplets, or by touching any surface, carrying the droplet, then touch your face, eyes, nose, mouth, or ear. WHO has stated that the incubation period of COVID-19 between 2 days to 2 weeks. SARS coronavirus 2 is a spherical, envelope, positive sense, single-stranded RNA virus of zoonotic origin. SARS coronavirus 2 contains 4 structural proteins, namely envelope, spike, membrane, and nucleocapsid. And they are responsible about virus binding, fusion, and involved in virus assembly during replication. The viral spike, as shown here, the viral spike glycoprotein binds to the human AC2 receptor present in the target cells in the respiratory tract. There are 12 functional open reading frames, which encode 16 non-structural protein on the SARS coronavirus 2 genome. Pathogenesis of coronavirus, especially the COVID-19, is divided into 3 parts, including viral entry and replication, cytokine storm, and coronavirus immune evasion. Regarding the viral entry and replication, after attachment of viral particles to the host cells, then a cascade of a process as entry, fusion, replication, and finally and release from host cell. The second step in pathogenesis is the cytokine storm. In case of SARS-CoV-2 infection, the main mechanism of acute respiratory distress syndrome is the cytokine storm, which is the uncontrolled deadly systemic inflammatory response resulting from the release of large amounts of pro-inflammatory cytokines and chemokines by immune effector cells. And finally, as in SARS and MERS coronaviruses, SARS coronavirus 2 can produce double membrane vesicles that don't contain pathogen-associated molecular pattern and begin replication in these vesicles. By this means, it avoids the host detection of their nucleotide. The spectrum of illness can range from asymptomatic infection to severe pneumonia and death. The most common symptoms in patients with COVID-19 were fever, cough, and dyspnea. Other new symptoms include loss of taste, loss of smell, and diarrhea, as shown in this figure. Individuals of all ages are at risk for infection and severe disease. However, the probability of serious COVID-19 disease is higher in people aged more than 60 years and those with chronic medical conditions like diabetes, hypertension, and cardiac disease. Virologic testing such as molecular diagnostic or antigen tests to detect SARS coronavirus 2 should be done in all persons with a syndrome consistent with COVID-19, while initial diagnostic tests for SARS-CoV-2 infection have relied on reverse transcriptase polymerase chain reaction platforms. More recent tests have included a variety of additional platforms. On the other hand, unlike molecular diagnostic and antigen tests for SARS-CoV-2 that detect the presence of the virus, serologic tests are intended to identify persons with recent or prior SARS-CoV-2 infection. They have been used as an additional diagnostic test in patients strongly suspected to have SARS-CoV-2 infection. The abnormalities seen in computed tomography (CT) of the chest also vary, but the most common CT images typically show bilateral pulmonary parenchymal ground glass and consolidative pulmonary opacity, sometimes with a peripheral lung distribution and a rounded morphology, as shown as the arrow pointed here. Currently, there are no FDA-approved drugs for COVID-19. However, an array of drugs approved for other indications as well as multiple investigational agents are being studied for the treatment of COVID-19 in clinical trials around the globe. The choice of what to do or not to do for an individual patient is ultimately decided by the patient with their provider. Some examples of these drugs include azithromycin. Azithromycin, it is an antibiotic approved by FDA for treatment of various bacterial infections. It was found that it can induces the interferon-stimulated genes and attenuating viral replication. While chloroquine and hydroxychloroquine are approved by FDA for treatment of malaria. They were found to increase endosomal pH and inhibiting fusion of SARS-CoV-2 and the host cell membranes and may interfere with binding of SARS coronavirus 2 to the cell receptor. Lopinavir and ritonavir are FDA-approved drugs for treatment of HIV. They can inhibit the 3 chymotrypsin-like protease in SARS-CoV-2. Remdesivir is an antiviral, but not approved by FDA. It can bind to the viral RNA-dependent RNA polymerase and inhibiting viral replication. It is mainly used in hospitalized patients with COVID-19 who require supplemental oxygen. There are insufficient data to recommend either for or against the use of COVID-19 convalescent plasma for the treatment. Researchers around the world are racing to develop a vaccine against COVID-19, with more than 170 candidate vaccines now tracked by the WHO. Vaccines normally require years of testing and additional time to produce at scale, but scientists are hoping to develop a coronavirus vaccine within 12 to 18 months. On the recent vaccine news, we found that to date, the only approved vaccine is by the Russian health authorities, which has yet to complete the clinical trials. Most transmissions are thought to occur through droplets, so face coverings, hand washing, covering coughs and sneezes, and maintaining 6 feet from others can reduce the risk of transmission of the virus. Without pandemic containment measures such as social distancing, vaccination, and use of face masks, pathogens can spread exponentially. This graph shows how early adoption of containment measures tends to protect the population. Thank you for your patience and attention.